my dear little cuties, I welcome you all to YouTube Brahmastra series. Today we are going to deal with sexual reproduction in flowering plants. Today is day 13 and I hope everyone is in loop. And one more very important thing, how are you all? I hope that everyone who is watching this particular video is doing absolutely good and you are having a fresh mind to study something new, that is this particular chapter. So I hope you are on this journey with me and I want each and everyone to be there from the start till the end of the video with me. You would understand things a lot better. You can ask doubts in the live comments. You know, if you have been there with me, you know that I definitely reply in the comment section. Okay, so you can ask me your doubts, you can tell me whatever you want in that. Okay, some ask for tricks and also, so I try to find certain tricks and put it there. But yes, you can also do the same. So be with me throughout the journey. It might be for 1.5 hours, but we would be completing everything that is needed for us. And I guarantee you that you will be getting how many marks? After being with me, 11 marks, total 11 marks for this particular chapter, you would be getting it. So there are 3 blueprints given by the government, my dear children, okay, according to blueprint 2, 1 and 2, you would be getting one 1-marker question, one 2-marker question, one 3-marker question and one 5-marker question. So all the type of questions would be there in the question paper from this particular chapter. According to blueprint 3, there will be 3 1-marker question. It can be MCQ or fill in the blanks, but mostly it will be MCQs only. Okay, then there will be one 3-marker question and one 5-marker question. There won't be 2-marker question according to this particular blueprint. So you can get any of this in on your day of exam. So we have to be prepared for all the type of questions, right? So accordingly, I have made something like this. I would be solving 10 1-marker question, 6 2-marker question, 4 3-marker question and 4 5-marker question. Why I have increased the number of 2-marker questions? Because when you study in parts, it becomes all the more clear. For example, I have 1 and 2 question, both of 2 marks each, but they could combine together and come for a 3-marker question. So I thought, why not explain you the concepts in bits and pieces so it becomes less boring for you, because I know when I explain a lot, you might get bored as well. So I don't want you to do that. You stay chill, okay? We are going to study. So I hope you're excited. If you're excited, let me know in the comment section. And this is the schedule, my dear children, okay? This is the schedule. You have to, according to this, you will be having classes. So if you want, you can take a screenshot. We are over here. Now from tomorrow, you would be having these many chapters. So if you're someone who want to complete all these chapters, you can follow this. Okay? Works? Chalo. The first question, let's solve it. Statement 1 says, apomixis is the production of seeds. Production of seeds from unfertilized ovules. So children, suppose this is ovule. Ovule will be known as seed. Ovule will be known as seed when inside the ovule, when the egg actually fuses with the male gamete. When this fertilization, when this particular process has happened, what do you call this? You call this as fertilization. And when this is happening, now you will call the ovule as what? The seed. But they are saying that apomixis is basically production of seed. You are basically producing seeds, but there is a twist in the story. From unfertilized ovule, this ovule is not fertilized. That means this fusion is not happening over here. Still, you are making a seed out of it. Is this possible? You might think it is not possible, but this is possible with the help of apomixis. So here, due to some unknown mechanism, what happens is that this particular egg, okay, this particular egg without fertilization transforms into zygote, and hence we say, since zygote has been formed, we say that the seed has been formed. So usually egg and the male gamete comes together, gets fertilized, that's when it forms zygote. But over here, the egg itself, somehow it is forming the zygote without fertilization. Now this process is known as apomixis. So apomixis is the production of seeds from what? Unfertilized ovule. So over here, you have the egg cell waiting for this, not sperm cell, we say in the case of plants, we call it male gametes, okay? The male gametes, but over here, without the male gamete coming and fusing, it forms the zygote and then it will form a seed as well. Okay, when it forms a zygote within the ovule, we will now call the ovule as seed. So yes, this is a true statement. Okay, how and all, it's a miracle that is happening within the plant's body, okay, but it does definitely happen because of certain hormonal things and things like that. Now, embryos produced from apomictic seeds are generally identical, are not generally identical to the parent plant. Now, they are saying that the embryo, zygote will form an embryo, right? They are saying that the embryo that is being formed, or basically the new plant that is being formed, it is not identical to the parent plant. This was a parent plant which was having all this thing. From here you got the embryo. It is saying the embryo won't be similar. Is it that so? Do you think it will be like that? The embryo would not be similar to the parent plant when the egg fuses with the male gamete. Now you have two sets of chromosome there. When you have two sets of chromosome, you are bringing out variation, and hence the embryo will be a combination of this parent and this parent. So it won't be exactly similar to this parent, it won't be exactly similar to this male gamete parent also. So basically the embryo will be kind of new. But over here in apomixis, where this fusion has not been taken place, where fertilization has not been taken place, the embryo will be the exact replica of the parent. Parent as in basically egg is given by the female parent, right? So it will be just like the female parent because no fusion has taken place. There is no sexual reproduction that is being taking place over here. This can be counted as a mode of asexual reproduction, or it can be counted similar to asexual reproduction. We say apomixis would give you clones, okay, similar to the parents. So here it has written that it is not generally identical to the parent plant. This is wrong. It is identical to the parent plant. So statement 1 is correct, statement 2 is what? False. Where? Yes, children, this came in model question 2024-25. Now, I want to tell you one more thing. If you are someone who know the answer already, you can tell me in the comment section, in the live section. I will be really happy to see you engaging in this particular series. Okay, you can pause the video also and you can try to solve the answers by yourself and do let me know. Okay, chalo. Next thing. Let's challenge ourselves. So, select the correct alternative from the choices given. Select the correct sequence of events in microsporogenesis. What do you mean by microsporogenesis? The formation of microspores. What does this microspores do, children? Microspores which are being formed, they are later transformed into what? The pollen grains. The pollen grains, and pollen grains are the one that will be having inside it. Inside the pollen grains, what you will be having? You will be having the male gamete. So since this is the guy that will be having the male gamete, I would be calling this guy as male gametophyte. The one that carries or basically has the male gamete, it will be known as male gametophyte. So pollen grains have it. So where did pollen grains come from? From the microspores. Now you are asked, what is this sequence of formation of microspores? There might be a lot of things at the end of which you get microspores. So what is the correct sequence of that? So let's see. The correct sequence will be sporogenous tissue, microspore mother cell, microspore tetrad, microspore. Is that true? Or is it this? How come you, how will you remember this? So basically we have to remember in order only. First comes S. So there are a group of cells, okay. There are a group of cells which forms a tissue which you call as sporogenous tissue. Okay, now this cells, each of these cells, okay, each of these cell, I will call them microspore mother cell. Okay, MMC, just like you have BBC, no, just like that, you have MMC, microspore mother cell. But each of the cells of this particular tissue are microspore mother cell. Now this microspore mother cell will undergo meiotic division. What will it undergo? Meiotic division. Now when one cell undergoes meiotic division, how does it look? Let's see. So over here we have one microspore mother cell. It undergoes meiotic division. You have two cells. Okay. Suppose it had 2n number of chromosomes. For example, let's take in the case of human beings, we have 46 number of chromosomes when we start. Okay. Now, in the first stage of meiosis happens in two stages. One is meiosis 1 and there will be meiosis 2 as well. In the first stage itself, the number of chromosome becomes half. That is, whatever number of chromosome is there is divided into half and given to both. So, it will become n over here. That is, if it was 46, it will become 23 over here. Half the number of chromosomes will be forming over here. Then again, this will undergo the second round of meiotic division. Do you think it will again half itself? No. Now it won't half. It will be just like this. So there will be n, there will be n. Here also it will be n, here also it will be n. But how many of you are getting, how many of them you are getting over here? 1, 2, 3, 4, ma'am. So these 4 things, no. What do I call it? I call it microspore tetrad. Tetrad as in 4. 4 of them are there. Now microspore tetrad will basically differentiate, move apart away from each other, and then would be giving you microspores. This microspores will be developing into pollen grains. This pollen grains will be having male gametes inside it later on. Okay, so this is the process. What is it? Sporogenous tissue you are having. Inside the sporogenous tissue you have mother cells, so many mother cells, one mother cell among them. Okay, I am talking about that. Microspore mother cell, which is over here, it will undergo meiotic division. There are 2 rounds of meiosis, meiosis 1, meiosis 2. Okay, in here you have 2n number of chromosomes, that is double the number of chromosomes that you usually having. What do you call this? You call this as diploid. Okay, you are making it half, now you call it haploid. So always in meiosis there will be half the number of chromosomes. Why do you think there will be half the number of chromosomes? Because you only think male gamete has half, female gamete have half, they both come and fuse, now you will have full. So n and n comes, you have 2n, so that's the original number. But if 2n comes, it will be 4n, double the number of chromosomes. We do not want that, which is why meiosis happens in all the gametic cells. So in here they are going to form the gametes at the end, hence meiosis has to happen over here. So you get haploid cells, again there will be meiosis 2, you will be still having just the haploid cells, and these cells will be known as microspore tetrad because 4 of them are there together. They will separate from each other, you get microspores, from microspores you get pollen grains, so hence this becomes the answer. Now I hope you would not be forgetting the sequence of what? The formation of microsporogenesis. Okay, micro is for male gamete. Okay, you are going to form male gametes in general later on. Okay, mega will be for females. Now the ploidy of perisperm present in the beet seed is, children, this is ploidy. I told you diploid as in double the number of chromosome. This is haploid, that means half the number of chromosome. So they are asking you what is the ploidy of what? Perisperm present in the beet seed. What is so specific about beet seed? So in beet seed, no, what happens is that generally, now we are going to talk about female part. Inside the female part, suppose this is the ovule. Suppose this is the ovule. Inside the ovule you have so many cells, okay, and you call this whole thingy which has the cells as something known as nucellus. What do you call it? Nucellus. And nucellus cell, one among them, for example this one among them will be later known as megaspore mother cell, just like how we had microspore mother cell. Okay, there we had sporogenous tissue, and all the cells of sporogenous tissue were able to form microspore mother cell. But here things are quite different. Not all the cells of nucellus will be forming megaspore mother cell, it will be just one cell. So this nucellus, okay, this nucellus, basically what happens is that is used up, it's somehow degenerated and things like that. When the megaspore mother cell basically will be forming megaspore, and then later will be forming something that is known as embryo sac, and in the embryo sac it will be having the female gamete. During the whole process now what happens is that in this particular nucellus, the other cells of the nucellus, it gets degenerated. But in some plants this nucellus, it stays just like that. It will be staying just like that, and it will provide nourishment. It will be providing nourishment later on through the, to what? To the embryo sac. To the embryo sac, which has been formed after the whole lot of process which has been happening over here. Okay, now they are asking, and this nucellus layer which is persisting, what do you call it? You will be calling it perisperm. This nucellus layer which persists is known as perisperm, and this is found in 2 plants which is given in your NCERT. One is beet, another one is black pepper. And now they are asking you the ploidy of it. Is it haploid, diploid, what is it? Basically the normal cells, it is just a normal cell, so it will be having 2n number of chromosome itself. It will be having 2n ploidy, that is it will be diploid. It will be diploid. So this is the answer. What about the megaspore mother cell? It will also be diploid. Once it undergoes meiosis, at the end you get microspore. Similarly, at the end you got megaspore. What is the ploidy of microspore over here? It will be haploid. Similarly here also it will be what? Haploid. It will be just having n number of chromosomes. Listen to me very carefully. I might be solving other questions also by telling you this thing. Okay. Now the embryo sac is also there. just like how you had pollen grains, I told you microspore was developing into a pollen grain. Similarly, one megaspore is going to develop into what? One embryo sac. And this embryo sac basically is known as, since it is going to have the female gametes, I will call it female gametophyte. What will I call it? I will call it female gametophyte because it will be giving me the female gametes later on. Okay, works? Works. So this will be diploid, my children. Please remember, it is persistent. This perisperm is found in beet and black pepper only. Okay. So I might be taking a lot of time, but I am explaining you certain things which is very much important. Okay. So I hope you are using it. If you think that I am wasting too much of your time, please do let me know. Okay. I just want to clear your concepts because this will be useful for you in the coming questions. Now, some plants such as Viola, Oxalis and Commelina produce two types of flowers. Namely, what are those type of flowers? Can you guess? Can anyone guess what are those two types of flowers? Basically, there will be one type of flower which will be having what say open, which the flowers will be open such that pollination can happen from outside, self pollination can also happen, other plants can also contribute their things. Okay, and there will be certain flowers which are always closed, so over here what will happen? If they are always closed, I cannot bring other pollen grains over here. Okay, so do you think that cross pollination or anything can happen? No. Here always self pollination would be happening. Here always self pollination will be happening. That is, for example, this is the flower. They have their own anther and things like that. Okay, they have their pistil, they have their pistil and they have their anther and filament and everything like that. From here to here things will be moving because it is a closed thing. Okay, so there always self pollination is only the option, but over here in the same plant itself you will be having flowers which are open. Okay, so there cross pollination and self pollination, anything can be happening over there. So what are these type of plants known as? Basically, what are this type of flowers known as? The open one, it is known as chasmogamous. Where is it? Chasmogamous. And the closed one, it is known as clistogamous. How can you remember clisto? Clisto has Cl at the starting, so closed. Okay, and chasmo as in open. Okay, so Viola, Oxalis and Commelina, all of them, they have these two type of flowers. The lower ones would be clistogamous and they will always do self pollination. They do not have an option. These guys can do self and cross pollination both. Cool. Chalo, next question. The correct sequence of embryonic development in angiosperm is. So, okay, I did something. Just a second. Okay. So, the next question. The correct sequence of embryonic development in angiosperm is. So, listen. Egg and the male gamete fuses to form what? Zygote. This zygote will now form embryo. But the embryo also has different stages. Okay, they are asking about that. What is the correct sequence of embryonic development in angiosperms as in the flowering plants? So, this thing you have to remember. Okay, first comes zygote. After the zygote comes something that is known as pro-embryo. Okay, where is it? Wait. I'll show from here. Okay, first comes zygote, then comes something which is known as pro-embryo. This thing that you see is known as pro-embryo. After that you have globular embryo which is like globe shaped. Okay, then you have heart shaped embryo. If you see, there's an opposite heart over here. There's a heart shaped embryo and then you will be having a mature embryo. Okay, so zygote, then what? Pro-embryo. Okay, then globular embryo, then heart shaped embryo, then mature embryo. Please remember, pro as in we say the initial portion is always known as pro. So zygote, pro embryo, then globular embryo does not have any shape, then it has a shape, that is heart shape, then it becomes mature. After it has heart, it gets heart broken. Later on, after that we say they have become mature. You can remember like that. So what is the correct sequence? Now can you tell me in the answers over here? It will be zygote, then it will be pro embryo, then it will be globular, then it will be heart shaped, and then it will be mature embryo. So this becomes your correct answer. Okay, cool. Chalo, next question. The most resistant organic material present in the exine of pollen grains is. What do you mean by exine? So pollen grain has three layers. One is its plasma membrane. One is its own plasma membrane. Second thing that it has is intine, which is the inner layer. Okay, and then it has a very strong layer, which is this. This is known as exine. Now I'll tell you one very important thing about exine. Exine is made up of one very resistant organic material such that this pollen grain can survive for a long time, and which is why it is used as fossils also. This is a very important question. Okay, so this exine is made up of what? Sporopollenin. It is made up of sporopollenin, which can sustain high acid level, alkali level and things like that. So good. It is so good. Okay. And since water and everything, some moisture has to be getting inside the pollen grain. So what do we have? We have certain space in between the exine. Because anyway nothing can pass through this hard surface, right? Because it has sporopollenin. So this space is known as germ pore. This is known as germ pore. And I'll tell you one more thing since we are talking about this. Intine, it is made up of cellulose. Intine, it is made up of cellulose and pectin. Okay, exine is sporopollenin, intine is made up of cellulose and pectin, and then inside that you have the plasma membrane. Okay, and this layer that you have over here is known as this pore that you are having over here, it is known as germ pore, which lets moisture and everything in, and also later for the formation of pollen tube you need this space over there. Okay, pollen tube, you will study about that. So this becomes your answer. This is 2023 March question. Next question. Tassels in corn cob represents what? Tassels as in when you are having corn cob, you might be seeing that yellow thingy that comes over from there. Okay, if you have seen corn, which does not like this, but yeah, anyways, they have certain things like this, right? What do you think it is? So they have covering and then they have certain things like this. What do you think it is? The tassels. The tassels in the corn cob, please remember, it is the stigma and the style. Stigma as in the one on which the pollen grains comes and lands, and style as in the one that carries the pollen tube and everything. Okay, so stigma, style and ovary, I hope you know, it's a part of pistil or the female reproductive system. Okay, stigma, style and ovary. So this tassels represents stigma and style. This is given in your NCERT. And what type of pollination does this show? Children, it shows wind pollination. They show wind pollination, which is why they have long stigma such that anything that comes through the wind can be caught by them. Cool? So next, endosperm persist in the mature seed in what? So what is endosperm, my dear children? First thing, what is endosperm? So basically, how would I explain? Okay, let me think. For that, we have to go over here. We'll go over here. Okay, and listen to me carefully. So what happens is that in the embryo sac, you have egg cell, and then you have two other cells which are known as synergids. And then over here, you have two nuclei. They are not cells. The two nuclei present, which is known as polar nuclei. Okay, even though it is shaded like this, it's not a cell that you have. They are two polar nuclei which stays in the middle. Okay, I'll zoom this and show you why it is not zooming. And at the end, you have three other cells which are known as antipodal cells. And now I will tell you the scenario. So this whole thing is the embryo sac which is there in the ovule. This whole structure is known as ovule. So your embryo sac, inside the embryo sac, lots of division happened, and that's when you get these 8 cells. You have 8 cells over here. Sorry, 7 cells over here. 1, 2, 3, 4, 5, 6, and basically the whole thing that is over here, it is known as a cell which is known as central cell. So this is a 7 cell thing. How many nucleus are there? Each of these cells have nucleus 1, 2, 3, 1, 2, 3. Now over here I said there are 2 polar nuclei in between, so there will be 2 more cells. So 8 nucleus is there and 7 cells are there. 7 cells are there and 8 nucleus is there. So what happens is that when the pollen tube comes, carrying along the male gamete, there are 2 gametes that it brings. Okay, always it will bring 2 gametes. So one gamete will be fusing with the egg cell. Another gamete fuses with these 2 polar nuclei. You have to remember this. First thing, egg cell and the male gamete. Second thing, the 2 polar nuclei and the male gamete. Now I can explain. Okay. Now we will go back to the question that we were having over here. Okay. So the first event, egg plus male gamete. This will give you the zygote. Now I told you the polar nuclei, and the male gamete. This is second event that is happening. Okay, there are two polar nuclei over here. This is the second event. So basically, what does it give you? There are two polar nuclei that is present over here. All of them, they have haploid number of things only anyways. So these two polar nuclei also will be having, for example, one polar nuclei will be having haploid, another one will also be haploid. Okay, now this male gamete will also be haploid. How many nucleus will be formed now? 1, 2, 3. It's not how many nucleus. Basically, how many chromosomes will be coming together? You will say now you have 3n amount of DNA, or basically chromosome, and this is known as triploid. This is known as triploid. Until now, we were talking about diploid, haploid and stuff like that. When the male and the female things come, what do you get? You get 2n number of chromosomes, that forms the zygote. Now, when these guys fuses, you have 3n number of chromosome, but what is the thing that is being formed? You get a big nucleus which is known as primary endosperm nucleus. Primary endosperm nucleus. Okay, you get 3 nuclei coming together, you call it primary endosperm nucleus, which is going to form the endosperm. And what is the endosperm? Okay, just like in how in human beings you have placenta for nourishment, just like that in the case of plants, to nourish the zygote, because zygote is going to form what? It is going to form an embryo. So to nourish that, you have endosperm being formed. So endosperm forms first, and then the zygote will be forming into what? Embryo. So in certain plants, this endosperm which is formed is completely utilized. They use it completely, and those plants which use it completely are known as dicots. They are known as dicots because they are using them, either using the complete endosperm, and they are going to make cotyledon out of it. They are going to make cotyledons out of it, and here they have 2 cotyledons. They have 2 cotyledons, do not get confused. They are using the endosperm and they are making something else out of it, which is known as cotyledons. So they have 2 cotyledons in them, which are known as dicots. For example, you have bean, you have pea, you have groundnut. All of them you can break it into 2, because these 2 things that you see over here are cotyledons. Now inside the cotyledons there will be some small thing that will be the zygote, or basically the embryo which is being forming. If you are providing enough amount of nourishment, water and all, it will sprout into a new plant. So basically these cotyledons are there for nourishment only, but these endosperms have been transported into this. Now there are certain plants which does not utilize full amount of endosperm. So they just make, instead of 2 cotyledons, they just make 1 cotyledon. They just make 1 cotyledon. That means the endosperm is still there. They are known as monocots. For example, you talk about grass, they come under that. You talk about wheat, you talk about maize, they come under that. You talk about castor, it comes under that. You talk about coconut, it comes under that. Okay, so basically castor is the one that has the endosperm still persisting. Okay, they have little amount of endosperm still left. For all of them, they have utilized it. Dicots utilize it. Okay, now I hope you will be able to solve certain questions, whatever comes out of this. Chalo. The residual persistent nucellus present in some seed is known as, when the nucellus is persisting in beet and black pepper, what do you call it? I have already explained. This is why I have explained. Okay, the different type of question comes, and if you do not understand the concept, you will not be able to answer the concept as well. So answer becomes what? Perisperm. It becomes perisperm. Cool? Don't write pericarp. Pericarp is something. Now, name the type of pollination that brings genetically different types of pollens to stigma. When it is genetically different type of pollens to stigma, that means the plant has to be different. If for example, this is the flower and this is the flower. Okay, over here also you have flower. Let me just change the color like this. It is different plant. This is same plant. From here to here, if pollen grains are coming, that means it is genetically similar. It's anyway from the same plant only. But from here to here, if it is coming, that means it has to be genetically different. This is parent 1, this is plant 2, okay, this is parent 2. So there are genetically different plants. So this type of pollination that happens from one parent plant to another parent plant, it is known as xenogamy. What do you call it? You call it xenogamy. From one different plant to another plant. They have to be of same species. That's the only thing. But anyways, they will be genetically different. For example, one human being, another human being, they are the same species, but they are different individuals. Now, when they come together, you get new combinations. You get it? So this is known as xenogamy. This came in 2023 July, and with this we are ending all the 1 marker question, and I hope I explained you well, okay, what all type of questions could be coming. And why did I explain? Because when I went through this chapter, I saw that there are repeated type of questions, but there are new type of questions which comes each year as well, which is why I want you to be really well prepared for this chapter. Cool? Chalo. So this is the thing that I was saying. When from one plant to another plant it comes, you call this as xenogamy. Okay, I'll tell you other thing also. When there is only one plant and from one flower to another flower things are coming, you call this as geitonogamy. What do you call it? You call it geitonogamy. That is, this is the flower, this is another flower. From here to here if things are going, this is known as geitonogamy. Now, suppose from this plant only, in there, with, for example, they have, this is the flower, it has the stigma, style and ovary, it also has the anther and filament. Over here, from here to here things are moving. This is self-pollination that is happening. This is also self-pollination because it is happening in the same plant, okay. But this type of self-pollination, where their own stigma is fertilizing their own thingy, sorry, their own anther is fertilizing their own thingy, you call this as autogamy. Self, totally self, 100% self. But over here, at least they are fertilizing the different type of flowers over here. So this is known as geitonogamy. Genetically, both of them will be producing similar things only. Genetically, the product that will be produced will be similar only because it's from the same plant anyways. So this is autogamy, this is geitonogamy. If you want a clear picture, look over here. Okay, so this is one plant, this is another plant. From here to here if it is coming, it is xenogamy. Now, from one flower to another flower of the same plant, what do you call it? Geitonogamy. From this flower to this flower itself, it is known as autogamy. Where is it? Yeah, this is known as autogamy. Now, autogamy will only happen when you have both the stigma, sorry, the stamen as well as the pistil over here. If you have both the stamen, that is a male reproductive system, and the pistil, that is a female reproductive system, then only autogamy can be happening over here. Okay. Similarly, geitonogamy will also be happening only if you have two different type of flowers over here. Either the flowers should be like this, where one flower should be having the pistil and the another flower should be having the stamen, or the flower should be like this, where it has both. That's only when geitonogamy can happen. So from one flower to one flower, that means from this flower to this flower things can move, okay, or probably from this flower to this flower things can move. This type of flowers should be there in them. They should be having both the sexes on the same plant. They should be having both the sexes on the same plant. The flowers can be different. You can have different sexual flowers. That is different. Okay, or you can have both the sexes in the same flower also. Do not get confused. Geitonogamy happens when, this is an important question again. Geitonogamy happens when flowers have, or basically I'll just write okay, plants have both the flowers, male and female, or when flowers have both stamen and pistil. That's when geitonogamy can take place. Cool? Chalo. We have completed that. Now we have to talk about the 2 marker questions. So let's start. Already we have invested too much amount of time for the 1 marker. Chalo. Question number 11. Complete the tabular column given below with respect to the male gametophyte of angiosperms. What is a male gametophyte? Gametophyte as in the one that will give rise to gametes. What is that in angiosperms? In the case of male, it will be pollen grains. I already told you. Can you try solving this? So what are the cells of the male gametophyte? Inside the male gametophyte, what are the type of cells that you could find? So you have 2 columns, you have to write like that. Then the shape of the nucleus of the cells. Can you try? Basically, I told you the gametophyte looks like this, where it has a plasma membrane, okay, and it has an intine which is made up of what? Tell me in the comment section. I already taught you. And the exine, which will be having pores like this. What is the exine made up of? What are these pores known as? Okay, anyways, so inside this there are two types of cells, children. The pollen grain, no, it undergoes mitosis and now it has two types of cells. Suppose this was a pollen grain, it underwent mitosis, two types of cells are there, and both the type of cells are here itself. One cell is like this, it occupies a lot of space, this much space. This is known as vegetative cell. It is known as vegetative cell, okay, and it has an irregularly shaped nucleus. What type of nucleus? Irregularly shaped nucleus. On the other hand, inside the cytoplasm, this is the cytoplasm of the vegetative cell. Okay, suppose this has to be the cytoplasm of the vegetative cell. Inside the cytoplasm of the vegetative cell, there is a dense cell found over here. There is a dense spindle shaped cell. This is spindle shaped cell, which is found over here, which has its own nucleus, which is kind of elongated, and also it has dense cytoplasm. Okay, this cell you will call it generative cell. What do you call it? You call this as generative cell. So how many cells do we have? We have vegetative cell and then we have generative cell. Okay, now this generative cell is the one that will divide further and give you one male gamete and the second male gamete. This is the generative cell that will be giving rise to the gametes later on, and this vegetative cell will be helping in the formation of pollen tube. What is pollen tube, children? Pollen tube is that something that will be carrying the gametes. So suppose it helped in the formation of pollen tube, and now the gametes which are being formed later with the help of this will be getting transported with the help of the vegetative cell's pollen tube. This pollen tube that is formed is because of the vegetative cell. And where is the male gamete going? Going to the female egg. Okay, it is going to the female egg cell. So that is the thing. So what the question is asked, cells of the male gametophyte is vegetative cell and generative cell. Shape of the nucleus of the cell, I told you in nucleus of the cells they are asking, not the shape of the cell. So in the vegetative cell, the nucleus is irregularly shaped. In the case of generative cell, they have not mentioned the shape of the nucleus. They have mentioned the shape of the cell, which is spindle shaped cell. So you can write, to be in the safe zone, spindle shaped cell, in bracket write elongated nucleus. The nucleus, we can say it is what? Elongated nucleus. Or probably, since they are asking about the nucleus, what you can do is write elongated nucleus and in bracket you can write spindle shaped cell. Okay, works? Cool? So your answer, this is how it looks and this is how you can answer. Cells, one will be vegetative and the other one will be generative cell. Over here you have irregularly shaped and this will be elongated and in bracket write spindle shaped cell. I think this would be doing. Works? Chalo, you got the 2 marks. Now differentiate between geitonogamy and xenogamy, we already said it. Okay, what do you mean by geitonogamy? When you have one plant, in one plant you have 2 different flowers. From one flower it is coming to different next flower of the same plant itself. It is known as geitonogamy. It will be known as geitonogamy. Xenogamy is when all together you have 2 different plants and from here to here the things have to be moving. It is known as xenogamy. Now the second difference will be the genetic difference. Okay, in geitonogamy, do you find genetically genetically similar products that will be formed? Yes. After fertilization, whatever you do, the plant will be genetically similar to the parent plant only, because anyways you are taking this fertilization is happening or pollination is happening between the flowers of the same plant. So whatever you do, the cells will be genetically similar. So the product will also be what? Genetically similar. But when you talk about xenogamy, here there is one flower. This is a different plant. this is a different plant with different genetics. So from here to here or from here to here, whatever happens, the product that will be found will be genetically dissimilar. It will be genetically dissimilar. I can say it will bring out variation. Okay, cool. Chalo. This is how you can write the answer. It is the transfer of pollen grains from the anther of one flower to the stigma of another flower of the same plant. If you are not writing this, you are losing marks. Genetically similar to the parent plant. Okay. Xenogamy means transfer of pollen grains from the anther to the stigma of a different plant. Genetically different from the parent plant brings variation. Okay. It will be bringing variation as well. This becomes your 2-marker question. Done? Chalo. This came in 2023 March. And this is a question that is being repeated also, so I will put star star star. What is Parthenocarpy? Give an example. So in apomixis, if you remember what I said, that was when the ovule. Okay, so I will tell you something about the female reproductive system. Over here, this is the stigma, this is the style, this is the ovary. Inside the ovary, you have something that is known as ovule. Inside the ovule, you have an embryo sac, if embryo sac is being formed over there, and you will be having the female gametes and stuff like that. You will be having the female gamete, the male pollen grain will be landing over here. Later, it will be forming a pollen tube, which will be carrying the male gametes over here. This is how things work. So usually, after fertilization happens, suppose male gamete and female gamete fused. What will happen now? What will I call this ovule as? Ovule will become seed. When ovule becomes seed, the outer layer that is there, that is ovary will become what, children? Fruit. Ovary becomes fruit. This is the usual case. But I am telling you, so until now in apomixis we learned it is a process where this fertilization does not have to take place. Still the female gamete will be forming zygote and will be known as seed. Similarly, in this, what happens is that without fertilization happening, this ovary is now going to make a fruit. Fertilization has not happened and this ovary will now make a fruit. And if fertilization is not happening, if I am saying zygote is not being formed, basically nothing is happening over here. Fertilization is not happening. Can I say the seed is not being formed? Seed is not being formed. Do not think it is apomixis. Apomixis is something different. Over here, I am saying fertilization has not taken place, but the ovary is making fruit. Okay, I didn't say that seed is still being formed. I am not saying. So these type of plants will be having seedless fruits. They will be having seedless fruits because nothing has happened over here. The female gamete is still just like that. So I will say it is seedless fruit. Nothing has happened. In the previous apomixis, the female gamete was forming zygote, but over here female gamete is not forming zygote also. Nothing is happening, so there is no seeds. But the ovary which is there outside, it is forming a fruit. This process is known as Parthenocarpy and this gives you seedless fruits. This is naturally occurring in the case of banana. This is naturally occurring in the case of banana. In some plants we induce it. And how does this happen, children? Usually because of certain hormones. Hormones such as auxin, gibberellin. These are the hormones. Because of some imbalance that is being created over there, the ovary will now become the fruit. Okay, and it can be induced, it can be made to happen, and for that we provide the auxin, we provide the gibberellin, and this happens in grapes. So we get seedless grapes, no? That is induced Parthenocarpy. That is induced Parthenocarpy, where we are inducing. So I hope you understood what is Parthenocarpy, where you are making ovary into fruit. There, apomixis, you are making ovule into seed without fertilization. Over here, we are making ovary into fruit and there is no seed. There is no seed. Example, you have this. So ovary develops into fruit without fertilization. Such fruit is known as Parthenocarpic fruit. This phenomena is known as Parthenocarpy, and example is banana, and you can give the example of grapes also. You can write induced Parthenocarpy. You can write it is because of hormones. It is because of hormones like auxin and gibberellin. Works? Chalo. Next question. Define the following terms: staminate flowers and pistillate flowers. Staminate flowers are the ones that will be having the stamen. Staminate flowers are the ones that will be having the stamen. What do you mean by the stamen? Stamen means you will be having a filament like this, and at the end of filament, what will you have? You have anther. In the anther, what is being formed, children? The pollen grains are being formed. Okay, so this whole structure, the stamen with an anther and filament. If a flower has this, you call this particular flower as what? Staminate flower. What do you call it? You call it staminate flower because it just has the stamen. So can I say it's unisexual? There is no other sex present over here. It's just the male part of the plant. So I will call this flower as what? Unisexual flower. It is a male flower. It is a staminate flower. Okay. Now you want pistillate flower. In the pistil, what do you need, children? For the pistil, you need ovary, you need stigma, you need style and you need stigma. Okay, it does not look like that. It looks like a funnel. So let me just make once more. So over here you have this, now you have this, then you have the ovary. So this is the stigma, this is the style, this is the ovary, and ovary has the ovule. Okay, now if a flower just has this, that is, it just has the pistil, you will call it pistillate flower. You will call it pistillate flower. So can I say this flower is also unisexual? It just has one. This is known as unisexual flowers. Okay, they are known as unisexual flowers. So this is the definition. Staminate flower, unisexual male flower bearing only the stamens. Okay, pistillate flower, unisexual female flowers bearing only the pistil. Okay, that is the thing. Now suppose you have a plant like that, where you have all the unisexual, okay, all of them are male flowers. All of them, they are male flowers. And another thing, all of them are the female flowers. What do you call this type of plants as? You call them dioecious. You call them dioecious. So in one plant there will be one sex only. Okay, example is papaya. Example is papaya. The question comes, in papaya, gytenogamy, xenogamy, which is possible? Do you think gytenogamy can be possible over here? They just have the male flowers. Do you think gytenogamy can happen over here? No. Is xenogamy possible? Yes. From here to here, things can get fertilization. So xenogamy is possible in dioecious plants. In dioecious, you will be finding only xenogamy. You will be finding only xenogamy, and the example is papaya. There is monoecious. Monoecious is when, okay, monoecious is when, okay, when you have the male flower, okay, and you have the female flower, or probably you have a flower that has both male thingy and also the female part. This is known as monoecious plant. So for them you have gytenogamy. For them gytenogamy is possible. From here to here things can move. Okay, then for them this is also possible. What do you call? Autogamy is also possible. From here to here things can move. But there are chances where you can have either these flowers or you will be having only these type of flowers. Okay, if suppose you are having just these type of flowers in monoecious, do you think autogamy can take place? No, autogamy cannot take place. So in monoecious again you have two things. In monoecious your flowers can be like this, where you have both, okay, or you have a flower which has both the things, that is also possible. So depending on that, autogamy will be possible. Okay, I hope you understood. Works? Chalo. Sporopollenin is one of the most resistant organic material known. Justify. How can I justify? Sporopollenin is an organic compound that can, it is most resistant, so that can survive in high alkali, high acidic conditions as well. This is how I can justify. Okay, which is why it is used as fossils also. Certain fossils are still being preserved. Certain pollen grains are still being preserved until date. From years they are still there because of the sporopollenin. So this is how I can answer. Sporopollenin is one of the most resistant organic material because it can withstand what? Strong acid and alkali. No enzyme that degrades sporopollenin is so far known. You can put this particular point as well. There is no enzyme that can degrade sporopollenin as of now. So because of this property, pollen grains can be preserved as what? Fossils. Sporopollenin is found where, children? It is found in the egg sign. 3 points you can write. Okay, you can make much more shorter points, but you have to write it. 2022 March question. Going ahead, differentiate between pericarp and perisperm. Perisperm I already told you. Okay, when you have certain cells, the nucellus, when it persists back, the nucellus which persists back and provides nourishment, you call it as perisperm. And where do you get it? You get it in beet, you get it in, sorry, there's only one E, okay? I mean there is only 2 E, I don't know why I wrote like that, okay. You can see it in beet and you can also see it in black pepper. Pericarp. Pericarp means basically, suppose this is the ovary, this was the ovary, right? And this ovary is now going to form a fruit. And there is ovule inside. This is the ovule. Ovule becomes a seed. Now, the outer covering of the fruit, the wall of the fruit, okay, the wall of the fruit is known as what? Pericarp. It is known as pericarp. And in some plants you might have seen, for example, if you take mango. So mango, the outermost layer, the whole thing, the layer of thing that is found in the fruit is known as pericarp, right? The outermost layer is known as epicarp, which is the yellowish thing that you find. Inside that you have some fleshy thingy. Fleshy thingy is known as mesocarp. It is known as mesocarp. Inside that you might find one hard covering, which is known as what? It is not the seed coat. Inside this you will be having a seed, which is the ovule, basically fertilized ovule, and the seed will be having its seed coat. That is separate thing. But this hard structure that you find is known as endocarp. Endocarp. So pericarp can be divided into epicarp, mesocarp and endocarp in certain plants. For example, mango. In other plants, for example, groundnut and all, you won't be able to find it like this. Okay, there you do not have fleshy mesocarp and stuff like that. So some plants have fleshy things. So there you can find these 3 different layers as a separate thingy. Other plant you won't be able to find it. So the outer layer, so basically this 3 layers that you find together, you call it as a pericarp. Okay, you will call this as a pericarp. So pericarp, the wall of the ovary develops into the wall of the fruit and is known as pericarp. The wall of the ovary was developing into the wall of the fruit, which is why it's known as pericarp. Epicarp, the outer layer, skin of the fruit. Mesocarp, middle layer, the fleshy in many fruits. Endocarp, inner layer, hard or membranous, surrounding the seeds. Okay. Perisperm is persistent nucleus, nucellus, not nucleus. Nucellus is called perisperm. It is found in black pepper and beet. And what does it do, children? You can write that also. It provides nourishment. It provides nourishment, and here it provides protection in some other ways, right? So you can write like that. Okay, cool. Next, so we're done with the 2 marker questions as well. Now we are jumping into the 3 marker questions. Let's see. Draw a LS of grass embryo diagram and label the following parts. So it's a grass. It's a grass, and grass is a monocot, as in it just has one cotyledon. Remember, it has one cotyledon. That means it didn't utilize the whole endosperm yet. Still it has some endosperm left and it will be having one cotyledon, my dear children. Cool. So basically, how can I make this? So in an embryo there will be always an embryonal axis which goes like this. Okay, there will be an embryonal axis which goes like this. And suppose you have one cotyledon over here. So I will make one cotyledon like this, and I will name it also. I will name it later. Okay, for your understanding, what do you call this? This whole thing that you see over here, this is an embryonal axis. Embryonal axis. Now, this is the one cotyledon that I am having. This is the only one cotyledon that I am having. This cotyledon has a specific name. I call it scutellum. This provides nourishment. This will be providing nourishment to the growing embryo. The embryo is here only. Okay, this is the embryonal axis. This is known as scutellum. Okay, now I told you little amount of endosperm is still been remaining. So I will make the endosperm right over here. So this is the endosperm that I am having over here. This is what the endosperm. I don't have to mention it, but still I will mention it. Now in the embryonal axis, okay, this upper region will be known as plumule because it is going to give rise to the shoot of the plant, okay, the stem and everything. This lower region is known as radicle. It is going to give rise to the root of the plant. Now the radicle is basically covered by the root cap. The radicle is covered by root cap. Obviously it has to go into the soil, so it has to be protected. Now plumule, no, over it also you will be having certain things like this, okay, which you call as coleoptile, and similarly you will be having certain cells over here. So I have to basically erase this scutellum from here and I have to put it somewhere else, but I will do that. Okay, so over here also I have some protection thingy like this, which is known as coleorhiza. This is known as coleorhiza. This structure you see over here is known as scutellum. So basically when the embryo develops, it will take the nourishment from the cotyledon, from the endosperm, and slowly will give rise to the plant over there, the roots over here. This is how things work, and this is the LS of, okay, longitudinal sectional view of what? The grass embryo diagram. So grass is a monocot. If you do not know it's a monocot, you will make a dicot embryo and come. So we want to make a monocot embryo and it looks like this. Works? Cool. Just give me a second, okay? A second. Okay, so in your NCERT you have a different diagram. If you want to practice that, you can practice that also, but I feel this is more easy for me to remember, and I think it will be easy for you to remember as well. You can draw like this as well. Okay, now if you want to go according to NCERT, please, okay, you can do one thing for once, try from NCERT as well. Since you know the concept now, since you understand the concept now, what you can do is, once you can have to try from NCERT. Once you have tried from NCERT, put down in the comment section that we have tried for LS of grass embryo. Okay, put down that we have practiced for LS of grass embryo. I will be too happy. Please do that for me. Okay, please practice it once. This question has been repeated so many times, which is why I am saying. So better practice from NCERT also once. If I can, in one short video, such as in reel or something like that, I can help you in the formation of that photo, that picture also. So if you want that particular reel, you can comment that also. Okay, either way. If you are not commenting, I won't be able to know that. That's why I am telling you, and it will stay in my mind if you comment in the comment section. Okay, otherwise in the live chat goes when we finish out, it is not reviewed later on, no? So I won't be able to remember it. So yes, draw a label diagram of transverse section of young anther. So anther, you know, it looks like this. So this is the filament, my dear children. Okay, this is the anther. You have 2 lobes in one anther. Okay, you have 2 lobes in an anther. If I come like this and I see what is the picture, it looks like this. So basically I have one lobe like this, and then I have another lobe almost like this. Okay, so if I say this is one lobe, this is another lobe. So it is why it is said it is bilobed. It is what? Bilobed. Now each lobe, if you see, it has 2 theca, 1, 2. So I will say it is dithecous. Now listen to me very carefully, okay? Over here I have to do something. The color has changed, but it's okay. I think you will be able to understand. Okay, anyways, chalo. First thing I will make is this thing. These have so many cells, my dear children. This thing that I am making has so many cells. Can you guess what is this? Can you guess what is this? This is your microsporangium. Okay, this is your microsporangium, or basically I'll call these tissues which is found over here, or the cells together, as porogenous tissue. So you have so many cells over here, so many cells over here. Okay, now outside this you have a thing to nourish, you have a layer that nourishes this porogenous tissue, that nourishes this porogenous tissue. What do I call this? I call this layer as tapetum. I call this layer as tapetum. Okay, it nourishes. Okay, it has more than one nucleus also. Please be remembering that also. It has more than one nucleus and cytoplasm and everything. It provides nourishment to the sporogenous tissue, because sporogenous tissue has all the microspore mother cells, which will be dividing to microspore, then to pollen grains and stuff like that. Now comes 3 layers which are there for protection. Okay, so first layer comes like this, which I call as epidermis. Okay, and now comes the next layer, which I should be coloring different color, right? So over here, suppose there is a next layer of cells. Okay, you call this next layer as endothecium. Epidermis, and then you have endothecium. If it is not visible, I'll write it clearly. Wait, with white only I write endothecium. Okay, and then there's a layer over here as well. There is a thing over here as well. This is the third thing that you find, which is known as the middle layer. It is known as the middle layer. So first epidermis, then endothecium, then middle layer. These 3 things that you see over here, these 3 things, 1, 2 and over here, they are all for protection, as well as whenever the anther undergoes dehiscence, that is, it breaks open to release the pollen grains. For that time also you need these 3 layers. So for dehiscence of anther and for protection, what do you have, children? You have these 3 layers. And then comes tapetum, which provides what? Nourishment. Then comes porogenous tissue, which will be having microspore mother cells. So this is how you can draw a label diagram of transverse section of young anther. I hope you understood the basic thing. Without understanding the concept, I don't think you will be understanding how to draw this as well. Works? Okay, this is a 3-marker question. This also has been repeated so many times. Please practice the diagrams. Okay, next question. Write any 4 characteristics of flower pollinated by wind. So flower pollinated by wind. So you only think, if a flower has to be pollinated by wind, that means the pollen grains which should be there. Okay, it should be traveling through the wind, right? The pollen grains should be moving through the wind. The pollen grains should be heavy or should be light? It should be always light, then only it can travel. It is heavy, it will sit down like that, right? So we need light pollen grains, and this pollen grains should it be sticky or not? If it's sticky, it just go randomly sticks at any place. It should not be sticky. What should be sticky? Your stigma should be sticky. The stigma who receives this, it can be sticky, but not the pollen grains. Pollen grains should not be sticky. Okay, now the stigma and the style should be very long so that it can catch the pollen grains which are moving around. So these are the few features that you are having. Apart from it also, you will be having so many other features. Okay, let's see that. Or for example, if there is only one flower means one stigma, one stamen, so that is a problem. But if you have multiple flowers coming together forming an inflorescence, this becomes easy, because any flower can catch hold of the pollen grain which is flying, right? So this is also one feature of, let's say, wind pollinated flowers. So wind pollination requires the pollen grains light in weight, okay? It should be non-sticky and powdery. It should be non-sticky and powdery so that it can be transported. They often possess well-exposed stamens as in long, tall stamens, okay? So that the pollens are easily dispersed into the wind current. Stamen, as in the male part, it should also be long and tall, so that the pollen grains which goes from the stamen, they can move longer distance as well. If they are short, obviously they try to move up, that won't be possible. It's against the gravity, right? So basically they should be tall enough. The stamen also should be tall enough. The flower must have feathery stigma. What it should have? It should be having feathery stigma. It should be having, where is it? Feathery stigma well exposed stamens, non-sticky and powdery pollen grains, lightweight pollen grains and things like that. Feathery stigma again, why? Because you want to catch them. So easily to trap airborne pollen grains. Wind pollinated flowers often have a single ovule in each ovary. So in each ovary they will be having a single ovule and numerous flowers packed into inflorescence, so that the chances of fertilization, pollination can increase. Okay, got it? So they asked for 4, I gave you extra 1 point also. Now what are false fruits? So what are true fruits? True fruits are the one where the ovary itself is going to become a fruit. False fruit is when the things apart from the ovary is now contributing to become a fruit. For example, if you see a flower, okay, this is the flower, this is how it looks. You have the ovary and this ovary is basically becoming the fruit. But in some type of plants, what happens is that these parts also contribute to become a fruit. For example, this is known as thalamus. This is known as pedicel. So this can also help in the formation of a fruit. So those type of fruits which are arising from other parts, apart from ovary, they are known as what? False fruit. Example, apple. Apple comes from the thalamus. Apple comes from the thalamus. You have cashew. Cashew comes from the pedicel. So over here, false fruit developed from parts other than the ovary like the thalamus or the pedicel. Example, apple. Apple from the thalamus, cashew from the pedicel, and you have strawberry also. These are examples of false fruits. So false fruits, Parthenocarpy, apomixis, these are 3 important terms which is given in your NCERT and you should be knowing about it. And I hope I have explained you. So whenever the ovary is not participating in the formation of fruit but other parts are, so just understand you're talking about this. Works? Now, describe the structure of dicot embryo with the help of labeled diagram. So over here we made the monocot embryo. Okay, where it has just 1 cotyledon. Now you have to make the dicot embryo. It's very fun. Let's make. Okay, let's make. So over here, dicot will also have embryonal axis, which goes like this. Okay, and it has dicot, that means 2 cotyledons. So 1 cotyledon will be over here, another one will be over here. This is the embryonal axis. The upper part of the embryonal axis is known as epicotyl. Epicotyl. The lower part is known as hypocotyl. Okay, the lower part from the cotyledons. Okay, if there is a cotyledon over here, the lower part from this is hypocotyl. From here, the upper part will be known as epicotyl. At the end of hypocotyl, you will be having your radicle, and radicle is covered by the root cap. Here also. At the end of epicotyl, you will be having the plumule, which will be giving rise to the shoot. And these are the 2 cotyledons which will be utilized for the formation of the embryo, for the development of the embryo. So now in the picture that is given in your NCERT, it looks like this, where the cotyledons, no, they are shifted up. So I can just change this and form something like, so from here they have made some structure like this. Okay, and now they have poured it down. These guys are down. So this is what it looks. So these are the 2 cotyledons. You have the plumule at the end, hypocotyl, radicle, and this. You get it? So it's just to make you confused. And describe the structure. Okay, if you have to describe it also, it consists of an embryonal axis with 2 cotyledons. This is the, where is it? This is the embryonal axis with the 2 cotyledons, and then the portion of the embryonal axis above the level of cotyledons is known as epicotyl. The portion above the level of this is known as epicotyl, okay, and which terminates with the plumule or the shoot tip. This terminates with the plumule, or what do you call it? The shoot tip. Now, the cylindrical portion below the level of cotyledons is known as hypocotyl, and it terminates in radicle, which will be in root tips, and then it is covered by root cap also. The root tip is covered by root cap. This is how you can explain, and this is how you have to practice also. So I hope you understood the basic. Again, when you see the picture, you might not understand. I hope that you have understood what I have tried to tell you. Works? Chalo. Now next question. Picture related to pollination is given below. This is a 5 marker, we will be doing it. So with this, we have completed the 3 marker as well. Until now, do you have any doubt? You can pause the video and ask me all the doubts. Okay, in the comment section, I will be clearing all your doubts. Okay, if you have anything, or if you have any request, or if you have anything good to say, definitely you can do that as well. Either way, your choice. Okay, so tell me that, and then we will be solving the 5-marker question. Now let's move on to the 5-marker question. So picture related to pollination is given below. This is the picture. Analyze the picture really carefully. There is a plant, there is something like flower or something like that, and there is an insect, okay, a bee. So will this pollination confirm fertilization? Do you think that this type of pollination or any pollination, it will guarantee that fertilization will occur? What do you mean by pollination? Pollination means transfer of pollen grains from the stamen, or basically from the anther to the stigma. Right, this is what pollination means. What do you mean by fertilization? Fertilization means male and the female gamete when they fuse together, that is known as fertilization. So after the pollen grains lands on the stigma, then there will be a pollen tube formation, then the male gametes will reach there and then fertilize. But can I guarantee fertilization every time whenever pollination is taking place? Can I guarantee this process will definitely happen? There is no guarantee for that. Okay, so I can say that pollination cannot guarantee fertilization. Fertilization means the fusion of male and the female gametes, okay, to produce a seed. And pollination means this thing, and you have to explain like that. Pollination is the transfer of pollen grains from anther to the stigma, whereas fertilization means fusion of male and female gametes. So every time when pollination happens, we cannot guarantee that fertilization would be happening. Okay. Now, what are the floral rewards provided by the plants to the insects to revisit? Now, insects are the ones that carries the pollen grains. So, plant have to do something. They are selfish. They want pollination to happen. If pollination is not happening, they are not able to reproduce and give out more progenies of their kind. So, they will become extinct. They do not want that. They want the bees to come over here, and hence pollination has to take place. So, over there, what does the plant give as a floral reward for the insects as a gift? It can be nectar or it can be pollen itself. It can be nectar, which they can use for their purposes, or else it can be pollen, which is highly nutritious. They can use that as well. Okay, so some pollen they will eat, some pollen will be still there on their body. It will just go on, brush around in other flower. It will might give that pollen off. They never know. Okay, so that is the nectar and pollen is one thing that you can write over here. Now mention the characteristics of flower in this pollination other than the floral rewards. What will be the character of a flower pollinated or basically insect pollinated plant? If I say that this particular plant is insect pollinated plant, basically what are the features that it will definitely have? First thing, it will be having brightly colored flowers, so that the insects, basically they cannot see all the colors, but they have certain things, urination, something like that, spectrums that works for them, and they would be able to see these colored, not exactly the color that we are seeing, but some other colors. Okay, so the flowers basically have to be brightly colored. Now if they are small, in size, they have to come together to make a larger group, which is known as inflorescence. So you would be finding flowers with greater inflorescence over there for the plants which are insect pollinated. Okay, so these are few things, and apart from that, if there is a flower that is pollinated by flies and things like that, it will give foul odor, because with the help of this foul odor, it could attract more and more insects. Okay, so that is also one thing. Rafflesia, which is the largest plant that we know, it has some very bad smell. Okay, which is how it attracts the pollinators, that is basically the house flies and stuff like that. So this type of pollination, do you think that pollination guarantee fertilization? No, it cannot confirm fertilization. Fertilization occurs when pollen successfully fertilizes the ovule, which may not happen in every case of pollination. Now, floral rewards provided by the plant to insects include nectar and pollen. Okay. Now, flowers, what are the features of them? Flowers are basically the plant. Okay. Flowers are large, colorful, fragrant and rich in nectar. When the flowers are small, several flowers are clustered to form an inflorescence, which makes them conspicuous, as in very large, very visible. Okay. Now, the flowers pollinated by flies and beetles secrete foul odors to attract these animals. Okay, so that is also one more feature. Works? Cool? Let's go to the next question. Explain the structure of megasporangium. So, megasporangium is a structure that will be giving you what? Megaspores. Right. So, how will you draw the structure? First of all, it's asking about explanation, but still you have to draw and show. How will you draw and show? So, basically suppose this thing that I am drawing is the ovary. This is what, children? The ovary. Now, inside the ovary, what do you have? You have something that is known as ovule. Agreed? Now, I am saying the ovary basically has a region like this. which is known as placenta. Okay, now this placenta is something that provides nourishment to the ovule, and it is attached via something that is known as funicle. What do you call it? You call it funicle. Okay, now the ovule that you see over here, it has two protective layers around it. So this is one which goes like this, okay, and there is one more which goes like this. And what do you call these layers as? You call them as integuments. You call them as integuments. Now, this region which you see, there is an opening, right? There is an opening over here. This is known as micropylar end, or this is known as micropyle region. Opposite to wherever you find micropyle, the opposite to it, you will be having the chalazal end or the chalaza. Please remember, this guy, this placenta and funicle can be here also. That does not mean that wherever funicle will be there, you will find chalazal. No. Opposite to micropylar end, you will be finding chalazal end. Okay. Now, inside this, so this structure that you see over here, this ovule that you see over here, from here to here, this ovule is basically none other than the megasporangium, because this is where the megaspore will be developing. Okay. In the case of Anther also, you saw it had microsporangiums, which was filled with what? Sporogenous tissue. Okay, it was filled with sporogenous tissue. Over here you have ovule, which is known as megasporangium, and it is also filled with certain cells which forms what, children? So I am making certain cells like this. It is going to form the nucellus. And one cell out of the nucellus will be what? Megaspore mother cell. Suppose this is the one. This one will be the megaspore mother cell. Okay, this megaspore mother cell, it will undergo meiotic division. Okay, so suppose this is the one, it will undergo meiotic division. It was 2n, it became n and n. This is meiosis 1. In meiosis 1, the diploid one becomes what? Haploid. After this, again it divides n, 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 n. Right, you get 4 of them. So what do you call? You call it megaspore tetrad or what? Will you call it megaspore tetrad? No, children, because what happens is that 3 of them which are formed, they degenerate. They get degenerated. One of them, it will be the functional megaspore. It will be the one that will give rise to, that will be giving rise to the female gametophyte, which is known as the embryo sac, which is known as the embryo sac. So basically this structure will now look something like this. This whole structure will look something like this, where there is embryo sac. This has now become an embryo sac where you have got one cell with one nucleus. This is the embryo sac. Now this nucleus undergoes division. This nucleus will undergo division such that how many nucleus you will get at the end? 1, 2, 3, 4, 5, 6, 7, 8. You will get 8 nuclei at the end. You will get 8 nucleus at the end. So how many times would mitosis have taken place over here with the nucleus? Once it divided, once the nucleus divided, gave rise to 2. Other time it divided, gave rise to 2. Other time it divided, gave rise to 2. So 3 rounds of mitotic division happens, which is when you get this. Now these nucleus are surrounded by cells. Okay. The 2 nucleus that you see over here, they remain like that, and you call these nuclei as polar nuclei. What do you call it? You call it as polar nuclei. In this, there will be one cell which will be known as egg cell, that is the female gamete, where the male gamete will come and fertilize it. Cool. So this is the diagrammatic view of megasporangium. Now I will show you how you should be writing the answer for this, how you can explain this. Okay, so this is how we need to write the answer. So yes, First thing you have to mention, ovule is what? A small structure attached to placenta by means of a stalk. What it is known as? It is known as funicle. You can see that right over here, placenta, and then it is attached by a stalk which is known as funicle. Next thing, the body of the ovule fuses with the funicle in the region called hilum. Oh, I didn't say that, right? So this particular region which is there, it is known as hilum, where the funicle attaches, okay, which appears as a small pore also. Okay, now the ovule has one or two protective envelopes known as integuments. You saw it over here. It has two protection which are known as integuments, outer integument and inner integument. Now, these encircle the ovule except at the tip where a small opening called micropyle is organized. So here, apart from this region, everywhere else you would be having integuments. So micropyle region will be always open, okay. Now, the chalaza is present opposite to the micropylar end, representing the basal part of the ovule. So wherever you find chalaza, that is the basal part of the ovule, even if it is round like this also, if micropyle is here, if chalazal end is here, still the basal part of the ovule will be towards the chalazal end, okay. Now, the integuments enclose a mass of cells with reserved food known as nucellus. Okay, this mass of cells with reserved food is known as nucellus. Now, the nucellus encloses the embryo sac or the female gametophyte. Nucleus will be, because you see over here, this is a nucellus, okay, and inside this one cell is dividing and doing all this thing. So basically your female gametophyte or embryo sac is going to form over here. You might still have nucellus left, some amount of nucellus left. Sometimes it gets degenerated off, sometimes it may persist. If it persists, what do you call it? You call it perisperm, right? Not perisperm. So this is how you can draw it and show. So this is the picture that is given in your NCERT, where you see hilum is over here. Hilum is mentioned over here. This is the funicle. So hilum, they have placed this particular thing over here. That's the only difference. So don't get confused. They have just placed this over here, which is why the funicle is here, hilum is here, and you have the integuments and everything. This chalazal point is still over here because micropylar end is over here. Okay? Works? So if you want, what you can do is, you can make the embryo sac over here. Okay, you can make the embryo sac over here. The embryo sac looks like this. I'll talk about this in the next question. So I hope this question is also understood, and this is a very, 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 very repeated question, my dear children. Please practice the diagram very, very carefully. Okay, so sometimes half of our work is done with the diagrams if you understand the concepts. So please try to remember the concept. Chalo. So next question. Picture of a mature angiosperm embryo sac is given below. This is second last question. Okay, this is what? An embryo sac. Now answer the following questions that follows. Which cells nuclei of the embryo sac produce zygote and the primary endosperm nucleus? Which among them cells or the nuclei produce zygote and the primary endosperm nucleus? So we need to find two things. Who brings zygote, okay, and the primary endosperm nucleus. So that is the thing. So zygote, I told you, suppose this was the thing. This was the embryo sac which was formed. In that, one cell was the egg cell. I think I need more space. How do I? Okay, so this might go when I erase it. Okay, this is the embryo sac. 1, 2, 3 cells are there. Okay, and I'm going to color the egg cell. This is the egg cell. Okay, these cells that you see over here, what do you call it? Synergids. Listen to me very carefully. I am going to explain you double fertilization as well. Okay, now 3 cells are over here. 1, 2, 3. These are known as antipodal cells. Antipodal cells. Now from here to basically here, it went. Wait. To explain you things, I might have to draw things properly. Okay, from here to here, this whole thing that you see, which is guarded by yellow color, it is central cell, and central cell has 2 polar nuclei. This is also known as the central cell. Now, the synergids that you see over here, they have something like this which is known as filiform apparatus. Filiform apparatus. So once the pollen tube carrying the male gamete comes, no, the filiform apparatus that you see, which would help, it would help guide the male gamete to go and fuse with the egg cell. So one male gamete comes and fuses with it. One male gamete comes and fuses with the egg cell, and this product is known as zygote. Now once this pollen tube enters like this, no, your synergids, they degenerate. The synergids, they are the good friends you are having over there. They degenerate, they die for you. So basically they are dying here for the egg cell. Now the next male gamete which is there, remember always there are two male gametes, okay. The next male gamete will be coming in and will be fusing with these two polar nuclei that you see over here. Okay, so what are you getting? So basically there is a male gamete plus you have two polar nuclei, and now it will produce something that is known as primary endosperm nucleus. What is it? It is known as primary endosperm nucleus. Fine. Now, listen to me carefully. These cells which were there, egg cells, antipodal cells, polar nuclei, all of them, they were haploid. They were what? Haploid in nature. All of them, children, they are haploid in nature. Why? Because you remember the one megaspore which was left, three of them degenerated, one megaspore which was left, it was the one which was undergoing mitotic division. The nucleus of it was undergoing mitotic division. So it had multiple nucleus, and then the nucleus was guarded by the cell membrane and things like that. That is how you got these cells. So all of them basically they are haploid in nature. Okay, and the male gametes which are coming, they are also haploid in nature. They will also be haploid in nature. So this guy gives you zygote, but zygote this time is what? Diploid. It will be having 2n number of chromosome. Over here the male gamete has n number of chromosome. The two polar nuclei, one had n, other one had n. So n, 1, 2, 3n is there. Usually you have 2n, that is why you call it diploid. Now this thing that is being formed will be what? Not diploid, but it will be something else. Something else, as in it will be triploid. It will be what, children? It will be triploid. Okay, so you have diploid thing, you have triploid thingy over here as well. Cool? Works? Now, listen to me. Why do you call this process as double fertilization? I know I am deviating from the topic, but it is very much important for you. Okay, so double fertilization because here one fertilization took place between egg cell and male gamete. Over here again one more fertilization is taking place between this guy and this guy. That is why it is known as double fertilization. Okay, twice fertilization is happening over here. Now, this thing that you see is known as triple fusion also. This thing that you see over here is known as triple fusion. Why? Because three nuclei are coming together, which is why it is known as triple fusion. And this thing that you see over here, it is known as syngamy or actual fertilization. This is known as syngamy or actual fertilization. Got it? Got it? So I hope you know the synergids and all of them, they are N. Now the question is, where did you get the zygote from? You got the zygote when the egg cell and the male gamete fused. The question is, where did you get the primary endosperm nucleus from? You got it when the male gamete fused with the two polar nuclei. Okay, works. Okay, now if you are asked about ploidy of any of these cells, what do you say? They are all haploid. They are all haploid. Once fertilization has happened, we say there is a triploid primary endosperm nucleus and there is a diploid zygote being formed. What is the use of this primary endosperm nucleus? This will be later giving rise to something that is known as endosperm. And endosperm is that something, as I said earlier, that something that provides nourishment for the zygote to form an embryo. So embryo has to be formed, right? So endosperm has to be formed before this thing happens because you are providing the nourishment, right? So which is why endosperm formation has to always precede the development of the embryo. It will come before the development of an embryo. And I gave you 3 questions answer, even though we didn't read the question, but I'm telling you. So which cells, nuclei of the embryo sac produce zygote and primary endosperm nucleus, children? So you know which cells, male gamete cell, Okay, embryo sac as in, so embryo sac, which cells of the embryo sac gives you zygote? The egg cell. Okay, primary endosperm nucleus, the polar nuclei, or what do you call it? The central cell. Okay, now what is the ploidy of antipodal cells and the primary endosperm nucleus? What is the ploidy of antipodal cells? Antipodal cells, it will be haploid. Okay, what is the ploidy of primary endosperm nucleus? That is the only one that is triploid. Okay, now why the endosperm development precedes embryo development? Because you have to provide nourishment, then only embryo can develop, right? So that is the reason. So zygote is produced by fusion of male gamete with the egg cell. The primary endosperm nucleus is formed when the second male gamete fuses with the central cell, which contains two polar nuclei in the embryo sac. Got it? Now the ploidy of antipodal cells. The antipodal cells typically have wait. What is written over here? The antipodal cells, okay, the antipodal cells have haploid. Please don't write diploid, okay, it is haploid. Now, primary endosperm nucleus will be triploid. Now, development of the endosperm precedes the embryo development because the endosperm is necessary to provide nutrients for the growing embryo. The endosperm forms first and stores food reserves that supports the developing embryo within, that it grows within the seed. So, that thing you should be knowing and remembering. Cool? Chalo. Now, we have the last question. Explain any 3 outbreeding devices in flowering plants. What do you mean by outbreeding devices? Look, inbreeding as in, for example, there is a plant, okay, in the plant you have several flowers. If breeding is happening in between these flower parts, okay, in between the same plant, you call it inbreeding, okay, it is known as inbreeding. Outbreeding as in, I am looking for some other plant, okay, fertilization is happening or pollination is happening between 2 different plants of the same species, that is known as outbreeding. Now, why do you think you need outbreeding devices? Because regular inbreeding, when it happens, no, the plants get something which is known as inbreeding depression. Okay, it's not depression as in the mental depression that we are having. No, it's separate, it's a different thing. Okay, so inbreeding depression means basically the plant might not be able to survive, they won't be able to naturally produce, or fertilization could not take place properly. This is known as inbreeding depression. So, to avoid that, the plant itself has come up with certain things. Okay, we are not doing anything, the plant itself have come up with certain things, and that is what they are asking you for. 3 outbreeding devices. 3 things that helps the plant to outbreed, not inbreed, to outbreed. What are those things? I'll tell you a clue and you tell me the other points, okay. One point is, basically, sometimes, for example, this is a flower. It has the female part and it has the male part. Sometimes this thing will mature later and this thing might mature early. So, probably when it gave the pollen grains, it is not still receptive to receive what the pollen grains, because it has not been matured. So, that is one way. Okay, that is one way. What do you think is the other ways for outbreeding? Can you come up with something? It's already there in your NCERT. I hope you have learned it also, but I still want something from you. Anyone in the crowd? Come on children, cheer up, you can do it. What are the other things that you can find? So over here, I'll tell you. Unisexual flowers. Why to do all this drama? Why not? Why not make unisexual flowers? As in, have flowers which will be unisexual. They will be having just pistillate flowers, such that, suppose a plant is having just pistillate flowers. So obviously you need pollen grains to come from other plant that has pollen tubes. Oh, sorry, not pollen tubes, pistils. Sorry. What do you call it? Stamen. Okay, you need other plant which has staminate flowers. So that is one thing. Have unisexual flowers. Okay. So unisexual flowers, if both male and female flowers are present on the same plant, such as castor and maize, it prevents autogamy but not gytenogamy. So there are 2 ways. Okay. Suppose in unisexual flowers, there can be cases where there is one plant, there is one flower over here, one is having the female part and one is having the male part. Okay. Still they are unisexual flowers only. It is unisexual flower because it has just one sex in one flower, but this plant is monoecious. This plant that you see is what, children? Monoecious. Okay, why do I say it's monoecious? Because it has two sexes in them. Separately, but yes, it has. So over here, geitonogamy is possible or not? Do you think geitonogamy is possible? Yes, geitonogamy is possible because it's happening in the same plant. Is autogamy possible? No, autogamy is not possible. This I explained it to you earlier also. Now, this is the case of what? You have to remember the examples. If both the male and the female flowers are present on the same plant, such as castor and maize. This is the case of castor and maize, and both of them, they are monoecious with unisexual flowers. Now, there is one more condition where there is monoecious, but they have bisexual flowers. Bisexual flowers, what do you mean by that? Basically, this flower will be having both. So over here, monoecious means the plant should be having both the sexes. Over here, the plant will be having both the sexes because the flower it has, right? So over here, if a plant is produced like this, then autogamy will be happening over here. Autogamy will be happening over here. Geitonogamy can also happen because the same flower like this will come over here. So geitonogamy will also be happening over here. So you have to remember that, okay? This is an extra thing that they are given, but you have to remember that. So basically, if you want to prevent autogamy in general, then there should be unisexual flowers, monoecious plants. Okay, this thing does not work. Now, male and female flowers are present in different plants, that is dioecious, that is another way. Okay, either have unisexual flowers, okay, so you can prevent autogamy. The next thing, but geitonogamy is not prevented, you have to remember that. Next thing, that is the better way is that have the plants in separate, have the sexes in separate plants. For example, dioecious plants, which prevents autogamy and geitonogamy. But does it prevent xenogamy, children? Does it prevent xenogamy? No, it cannot prevent xenogamy by any chance. So papaya is an example of dioecious plant. Okay. Now, dicogamy is there. This is a technique. In some species, the pollen release and the stigma receptivity are not synchronized to prevent self-pollination and favors cross-pollination. This is what I was saying. So, pollen release time and the stigma receptivity time is not synchronized. If the stigma is not receptive, it won't be able to get the pollen, and hence the fertilization process won't be happening. So, this is known as dicogamy. Okay. Next one is known as protoandry. Okay, what is known as? It is known as protoandry, where what is happening? Flower in which anther mature earlier to stigma to prevent self-pollination. Over here, in the flower, what is happening? The anther is maturing first. This guy is maturing first. Okay, this is known as protoandry. Okay, and there is protogyny also. Okay, where the pistil will be maturing first. That is known as protogyny. I hope I am messing with the spelling. Yeah, where is it? Yeah, protogyny. Okay, where in the flower the stigma matures earlier to the anther to prevent what? Self-pollination. Then there is something that is known as self-sterility or self-incompatibility, which is totally genetic. As in, for example, they are receptive also. Still, when the pollen comes and lands over here, it won't be able to make the pollen tube, because there are some genetic changes that is happening, which is why it won't allow the formation. It won't allow the formation of pollen tube, and this is known as self-sterility. It is sterile. It is not fertile, it is sterile. It is not going to go ahead with anything. So, self-sterility or self-incompatibility, the pollen grains do not germinate on the stigma of the same flower due to mutual inhibition. This is genetic mechanism that prevents pollen germination on the stigma. Okay, so what all do you have? You can either make unisexual flowers, or probably you can have both the sexes in different plants. For example, dioecious thing. Over there you can prevent autogamy and geitonogamy. Okay, and then dicogamy is there, where basically stigma receptivity and the pollen release is not synchronized. Protoandry is there, where basically what happens, the anther matures first. Okay, protogyny is there, where the the pistil that will be maturing first. Then there is self-sterility or self-incompatibility, that is a genetic mechanism. Okay, so these are the ways where things work. Okay, so that the plant supports outbreeding rather than inbreeding. So they were asking for 3. So I will suggest whenever they ask for 3, go for these 3. 1, 2, and then 3. Okay, that will be the best one. So you have something to explain when it comes for a 5 marker. I hope you understood that. So with this, children, we have completed all the questions that have come okay, repeatedly, and I would love to request you that please go throughout your NCERT, practice diagrams. It is really important. If you're messing up with the diagram, you might mess up with explanation also. So try to understand the concept and then go with the diagram. Okay, or probably with the diagram itself try to understand the concept. I hope I was able to explain it to you very well. If not, do tell me, I will improve myself. Okay, and if I said nicely, if you have understood a lot of things, then definitely do let me know that as well. Okay, so this chapter is really important. Just go through it during exam time in 2x speed. It will be really helpful for you. That's my guarantee. Okay, and that's all. So that's all. If you have any queries or anything, you can tell it to us in the comment section, or you can put it in the live section format also. Okay, and if you want the PDF for the same thing that I did, you have to fill in the form that is given in the description box. If you have do not find in the description box, tell it to me, I will give it to you. Okay, done. And if you like the video, do not forget to subscribe to our channel and put down your cute little comments. We would love to read that. And all the very best. Please be tension free. You are going to do the best. So bye bye, take care.